Awesome. Well, good morning, everyone, and welcome. Thanks for joining us today for our monthly consortium webinar series. The title of today's talk is going to be on evaluation of soil and plant health in crops irrigated with oil field produced water in the Permian Basin. So my name is Zach Stoll. I'm going to be serving as our MC for today. Before we get started, just a couple of housekeeping notes. This session is being recorded, as you had heard. It's going to be shared with everyone after. You're welcome to ask questions at any time using the Q&A function and the chat function within Zoom. And for recording purposes, we just please ask that all guests turn off their video until the Q&A session. If you experience any technical difficulties, use the chat and our lovely admins, Amanda and Rosa, will gladly help you out. And so with that, I'm thrilled to introduce our speaker today. So joining us is Huma Tariq Malik, who is a PhD candidate and a Fulbright scholar in the soil and crop sciences at Colorado State University. Her research explores sustainable water management strategies with a particular emphasis on reusing produced water in ag. She is investigating how this practice influences soil health, productivity, and the soil crop microbiome with the goal of developing solutions that balance agricultural needs with environmental sustainability. So that was a really impressive intro. So I'm just going to turn it over and please join me in welcoming Huma. Huma, the stage is all yours.
Hello, everyone. Good morning. Thank you, Zach, for the introduction. Today, I'll be sharing our work on the evaluation of soil and plant health in crops irrigated with oilfield produced water in the Permian Basin. So produced water, I know it's a produced water consortium, but still wanted to share a brief description. Produced water is the water that exists naturally in the oil and gas formation. So when the wells are drilled to extract oil and gas, the water comes up to the surface along with it. It's the largest byproduct of oil and gas production, and while it was traditionally treated as waste, it's increasingly being studied as a potential alternative water source, especially in water scarce regions. So every year, the United States generates around 900 billion gallons of produced water, massive amount. So to give you a sense of scale, that's equivalent of about 1.36 million Olympic-sized swimming pools. And on average, for every 1 barrel of oil, about 7 barrels of produced waters are brought up to the surface as well. Produced water is a chemically complex mixture that typically contains surfactants, biocides, polycyclic aromatic hydrocarbons, salts, metals, various petroleum hydrocarbons. But from an irrigation standpoint, it's also important to note that it can contain crop nutrients like nitrogen, phosphorus, and potassium, and also boron, which at elevated levels can be harmful to the crops. So composition of produced water varies significantly. Based on multiple factors. It can be geological formation, reservoir conditions, chemicals added during the drilling process, drilling techniques. So, for example, this map here shows total dissolved solid, which stands for which vary among petroleum reservoir in the US. As you can see in the map, TDS values are relatively low in many areas in the West, like Colorado and Wyoming, but in some places it can exceed about 400,000 milligram per liter. And it's just one example. And just to give you the context, TDS of the ocean is around 35,000 milligram per liter. So it can be many folds greater than that. And TDS is just one value. The other components can also vary significantly, like metals and hydrocarbons, etc. So the most common techniques for produced water management are underground injection, which accounts for 85% of the volume. The rest, 13%, is the treatment, and less than 2% is used for the reuse. So why we need to explore treatment options beyond oil and gas field? So first, the volume of produced water generated far exceeds what the oil and gas industry can use internally. So the question becomes, what do we do with excess? Currently, the main disposal method is deep well injection, which has been linked to induced seismicity, in layman terms, man-made earthquakes caused by injecting large volumes of water underground. In some cases, when water is injected into shallower formations, we also see this phenomenon called bulge in the earth and blowout phenomenon, which can cause surface damage and safety risks. So because of these challenges, an unprecedented need for adequate management and reuse solutions has emerged. So 80% of produced water is generated in the Western United States. So the amount generated can vary by location, but in some cases can offer substantial water resource for the area. So in many places in the West are arid communities, so which often have a desire to reuse this water if possible. So on the lands west of the 90th meridian, which is the red dashed line shown on the map, operators can take advantage of NPDES permits, which are National Pollutant Discharge Elimination System permits, which allow them to release produced water for agricultural purposes if it's of good enough quality. So can produced water be reused for agricultural irrigation? It appears to be a very promising opportunities for the reasons I've discussed in the previous slide. But untreated produced water contains contaminants of concern from an irrigation standpoint. So therefore, in-depth analysis of produced water's effects on soil and crop health is essential to help guide treatment requirements. And to address this need, our group at Colorado State at Bork Lab, we have conducted a series of studies, and I will walk you through some of those findings very briefly. So there's this one study where we looked at what happens when wheat is irrigated with selected hydraulic fracturing chemicals. So what we found was that some of those chemicals were actually taken up into the grains and stem of the crop. So the key takeaway is that produced water must undergo treatment before food crop irrigation. And this was just an academic exercise. Obviously, in real life, no one's going to directly irrigate the crops with hydraulic fracturing chemicals, but we just wanted to see the uptake potential of those contaminants. So there was another study where we evaluated the effects of produced water irrigation on soil and plant health. So even when we diluted the produced water down to 5%, so 95% was tap clean water, and then 5% was produced water. Still, we saw decrease in soil health, wheat yield, and microbial diversity, likely due to the variety of contaminants in the produced water. So with another study, we found that produced water irrigation increased pathogen growth on the crops irrigated with produced water as compared to the ones which were irrigated with tap water and salt water. So produced water did lead to the suppression of plant immune system. So then there's another study we published this year where I served as a lead author, where we conducted comprehensive analysis of low salinity conventional produced water's effect on crop agronomic traits, soil health, soil metabolome, soil microbiome. So we went all in with this study. And conventional produced water, because it is extracted from sandstone, offers potential for irrigation because of lower salt levels and toxin levels most of the times. So we wanted to see that when we use untreated, low salinity, low toxin water by employing multiple analytical approaches, will we be able to detect early warning indicators of environmental stress or not? So what we found was that, and we used three irrigation regimes. We used tap water, clean tap water. Then we used 50% diluted produced water, and we use undiluted produced water. So results showed that while wheat grain metabolome concentration, above ground biomass, plant height showed no significant differences across treatments, grain yield was highest in the produced water, undiluted produced water treatment, likely due to the presence of nutrients. However, even that low salinity produced water increased the soil salinity, pH, which was accompanied by a decline in aggregate stability. And microbial communities in produced water irrigated soil also demonstrated functional shifts, including greater number of hydrocarbon degradation genes. So these subtle ecological changes offer insights into long-term implications of reusing insufficiently treated produced water, even though it was low salinity water. So while we have done all these studies, bunch of these studies in the greenhouse, why we need to go out in the field? Because greenhouse studies provided controlled insights into produced water irrigation, provided us with the baseline, but they can't capture field scale variability, soil, climate, management practices. So field studies allows us to see the real time impacts of, so allows us to use produced water in real time as well. And it's critical to assess impacts under realistic conditions and provides evidence for practical reuse potential and management requirements. So that's why we decided to do this irrigation trial in Orla, Texas, in Permian Basin. So the Permian Basin alone, operators produce about 3.93 billion barrels of produced water in 2019. So managing that volume is a major challenge. Most of it, as I discussed before, goes to deep well injection, which can contribute to induced seismicity. But there's also an opportunity here. After meeting oilfield needs, roughly around 2.76 billion barrels could be treated for beneficial reuse. And this treated produced water can help stretch scarce agricultural water supplies. So effective treatment and monitoring can convert this challenge or problem into supplemental water resource. So for this project, it was a massive project. Can divide it into three parts. So first was produced water treatment, and Dr. James Rosenblum from Colorado School of Mines was overseeing this part. And then chemical and toxicological characterization of produced water. And then we at Colorado State led the agricultural evaluation of the crops irrigated with the produced water. And our motivation, if I can describe it plainly and simply, it was just to see that can produced water be treated to quality fit for sustainable reuse in agriculture. Objectives of our study was to evaluate the feasibility of using produced water for irrigation in the Permian Basin, assess impacts on soil health and crop performance, and to characterize changes in soil microbial communities under produced water irrigation. This is our field plot in Orla, Texas. So we first excavated this quarter acre pit, lined it with black geomembrane to prevent any seepage, and then backfilled it with the soil. So for the treatment, this is an overview of the treatment train we use for produced water treatment. So we started with the raw water at about 110 grams per liter TDS. First, the pre-treatment was performed. Then water was passed through the membrane desalination, and this step strips out most of the salt, and the reject stream become brine concentrated to about 200 gram per liter. And then the permeate moved forward to the next step. So our overall water recovery was 40%. So for, let's say, every 10 barrels in, 4 barrels become product water and 6 were brine that must be managed. And finally, the permeate from membrane distillation passed through the polishing step resulted in fresh water about 600 milligram per liter TDS, which is well within the irrigation guidelines. So this table gives the overview of the quality of raw produced water versus treated produced water. So our raw produced water started at the pH of 6.84, which after the treatment, it was at about 7.76. Alkalinity at the starting point was 410, which was brought down to 225. TDS, which was around 12,000 milligram per liter, was brought down to 642 milligram per liter. Same goes for total organic carbon, total nitrogen, and chloride. It was reduced significantly, about at 99% removal. So there are some additional water quality data that could be of interest of any of the audience members. So our treated produced water contain around 10 milligram per liter of ammonia. That's higher than typical background in irrigation supplies, but not a concern. Sometimes farmers, growers use even higher amount for fertigation. So same goes for nitrate. 0 to 5 milligram per liter is typical of clean irrigation sources. Arsenic is well below irrigation guidelines, and then we did not detect any diesel range organics and gasoline range organics, which was a huge success for us. Same goes for gross alpha and beta, were at very low ranges, less than even the drinking water guidelines. So the pit, line pit I showed you earlier in this one of the slides. So we filled it back with the soil and then divided into 4 quadrants using this waterproof barrier. So we didn't want any cross contamination from either of the quadrants. So 2 of the diagonal quadrants we use for freshwater irrigation, and the other 2 we use for produced water irrigation. So our experimental setup, we use water sources. One was treated produced water from Permian, and then we use municipal drinking water. The reason for using municipal drinking water was because the local, we wanted to use the local fresh water, but it was too saline, I think about 4,000 to 6,000 milligram per liter TDS. So we wouldn't have any experimental design to compare treatments with if we were to use that water. So we ended up using municipal drinking water. And we used drip irrigation system and installed soil moistures to track it remotely as well. We use the crop we used was sorghum Sudan grass, and we choose sorghum Sudan grass for 2 main reasons. First was timing. We started this experiment relatively late in the season, around July 22. So it's a fast-growing summer annual that can establish well under those conditions. And secondly, it is known to be a tolerant crop, so particularly to stresses like salinity and boron, which made it a good candidate for initial testing, because then using a resilient crop allowed us to evaluate soil and microbial responses without the experiment failing due to crop sensitivity. Yeah, and then the total duration of the experiment was 97 days from planting to harvest. So as with the field experiment, things sometimes can go south. So one of the plot, which was produced water 2 plot, was due to the broken irrigation line. It got flooded a couple of days before the sampling or harvest. And because water kept standing there for a couple of days, so it created saturated condition that usually limits oxygen in the soil. That affected a couple of parameters, which I'm going to show later in my slide. So this was our plot 77 days after planting with luscious green sorghum Sudan. And at this point, we were not seeing any signs of stress or any differences between produced water irrigated plots and freshwater irrigated plots. So then by the end of experiment, the field plot that I showed you earlier, each quadrant was divided into a grid of 3 by 3 foot squares using ropes. And from this grid, 5 3 by 3 foot squares were randomly selected for further sampling. And we sampled leaf samples for phylosphere microbiome analysis. We collected bulk soil core samples for microbial analysis, and we also collected rhizosphere soil sample for microbial analysis. And rhizosphere is the soil which is kind of attached to the roots or closer to the roots. Then we collected bulk soil sample for general soil chemistry and physics as well. And by the end, we collected whole plant samples for agronomic traits analysis, which was biomass and yield. So this figure here shows biomass, above ground biomass differences between the 4 quadrants. So on the y-axis, on the x-axis we have all the treatments, and on the y-axis we have the value for above ground biomass. And the letters or alphabets above the boxes denote statistically significant differences between treatments. So groups sharing at least one letter are not significantly different. So when we looked at the 4 plots individually, we saw no significant differences. And then we combined the data from 2 of the freshwater plots and 2 of the produced water plots, and we saw no significant differences. Same goes for plant height. So with plant height in 3 of the quadrants, we did not see any significant differences. But freshwater 1 plot, the plant height was significantly greater than all the 3 other plots. And one of the things we noticed that because we use drip irrigation, and this plot was closest to the irrigation source, and over the crop growth cycle, we noticed some loss of pressure in the further away plots. So could be due to that those plots were not getting, were getting maybe little less amount of irrigation water than the FW1. But when we combine the results from both the freshwater quadrants and the produced water, there were no significant differences between treatments. So we also analyze metals and metalloids in the soil. For calcium, among all 4 treatment, we did not see any significant difference between the plots and the treatments. Cadmium, sorry. Same goes for lead. We did not saw any significant differences between the treatments. Same for arsenic and chromium as well. So for major salinity contributing ions, sodium, there was some variability, but it was not significantly different between all 4 quadrants between freshwater and produced water. Same goes for chloride. We did not see any significant differences between treatments. Same for calcium. For magnesium, we saw that freshwater quadrants had significantly higher magnesium than produced water, and then we realized that it was due to the higher amount of magnesium in the freshwater. For macronutrients in the soil, we did not saw significant differences between nitrate, between the treatments. Same goes for phosphorus, potassium, and sulfur. We did not see any significant differences. For micronutrients, with copper, we did not see any significant differences between treatments. Same goes for zinc. We did not saw any significant differences. For iron and magnesium, for produced water 2 plot that I mentioned earlier was flooded a couple of days before sampling. So what happened here, what we think is that the broken irrigation line caused water to stand for a couple of days before sampling, which created saturated condition that limited oxygen in the soil. So this causes the soil environment to shift from oxidizing to reducing condition, which likely increase the solubility of iron and manganese. It's important to mention that we use the DTPA method of extraction that does not reflect the total iron and manganese, but the portion that becomes more soluble and extractable under a reducing condition. So while total iron and manganese may not have changed, only the soluble fraction was higher due to anaerobic environment. For ammonium in soil, which was one of the most important element of interest here. Because our produced water contained little bit higher ammonia. Soil ammonium at harvest did not differ among treatments because ammonium is highly transient, so it's rapidly nitrified to nitrate and taken up by plants or leached. And the added N load was very small relative to the soil pool, so we wouldn't expect any increase in ammonium by the end of the season anyways. So this figure shows boron under both irrigation regimes. Um, Boron was significantly higher in the produced water treatment, and which was expected because it was higher in the treated produced water as well. Boron is a unique element because it's both as an essential nutrient and a potential toxin. So crops need it in a very small amounts, about 0.5 to 1 milligram per kilogram of soil. Below that range, plants suffer deficiency, but above it, boron becomes toxic. And then toxicity shows up as leaf burn and stunted growth and yield loss. But in our study, because we use sorghum Sudan grass, which is more tolerant to boron than many other crops, we did not see effects in crop agronomic traits like yield and biomass. I also want to reiterate that while sorghum may not immediately show severe toxicity, long-term accumulation or planting of more sensitive crops in the same soil could result in significant yield losses. And this makes boron one of the most important elements to monitor while evaluating produced water reuse. So we also analyzed soil biological properties. We looked at soil organic matter under different irrigation treatments. There were no significant differences between freshwater and produced water treatments. All soil remained around 2% organic matter, which is typical for the area. And we also looked at PMN, which is potentially mineralizable nitrogen. And we did not saw any significant differences. And this PMN, it usually reflects the amount of organic nitrogen that can be converted into plant available forms by microbial activities under favorable condition. So higher PMN generally suggests better nitrogen supplying potential and more active soil biology. Then we also looked at soil respiration. Again, we did not see any significant differences, and we use it as a proxy for microbial activity. So it measures the amount of carbon dioxide released by soil microbes as they break down organic materials. There is very little variability among replicates, but there were no significant differences between freshwater and produced water. We also looked at soil chemical properties. So with pH, there were no significant differences between treatments, and it fall within a narrow range of around 7.9 to 8. So produced water irrigation did not alter soil pH compared to freshwater controls. So now this figure shows soil electrical conductivity under both irrigation treatments. So interestingly, the produced water treatments did not increase salinity compared to freshwater. The lower EC we observe in produced water 2 may not be due to irrigation water itself. Again, that plot was flooded, and which likely leached soil downwards and diluted it, so lowering the EC readings. For microbial sampling scheme, we follow the same sampling protocol I explained earlier, that we sampled we collected 3 samples from each 3 by 3 square, and then composite it. And for each quadrant, we had 5 samples. For bulk soil, for phylosphere, and for rhizosphere as well. So minor shifts in microbial communities across bulk, rhizosphere, and phyllosphere soil compartments were observed under produced water irrigation. In both rhizosphere and bulk soil, treated produced water resulted in increase in nitrifiers, which is potentially due to the elevated ammonia in the water, in the produced water. And then there were no detectable differences in fungal community composition that we observe. So overall, the minor shifts we observed were likely due to elevated ammonia and boron. So from the data we have analyzed so far, we can conclude that treated produced water irrigation showed minimal overall impacts on soil and crop health. However, some parameters such as boron and ammonia were elevated, so that suggests that treatment system could be further tuned or refined. But I want to emphasize and reiterate a very important point that in-depth chemical and toxicological assessment of treated produced water is essential before ever using it for the crop irrigation. And because this was just one growing cycle, though we saw promising results, but future studies should evaluate irrigation at larger field scale across multiple growing cycles and include different crop and soil types. So our final conclusion that our study indicated that produced water can be treated to allow for successful crop irrigation. And we do not necessarily need to grow food crops with produced water. There are other potential uses as well. So let's say in Orla, we began with this heavily disturbed, barren plot land in the middle of nowhere, which over time transformed into these lush green fields. And by the harvest time, when we went there, we were surprised that it attracted some wildlife. There were birds and insects creating a vibrant habitat. So it can be used for revegetation of disturbed lands to support native grasses and plants. In oilfield reclamation and rangeland restoration, can be used for wildlife habitat creation to sustain vegetation that attracts birds, insects, and pollinators, and can also be used for ecosystem services like reducing erosion, improving soil cover, contributing to carbon sequestration. So this project was a massive undertaking. So we had multiple industry and academic partners. We at Colorado State University oversaw the agricultural research design, planting, irrigation, and soil analysis. Our colleagues at Colorado School of Mines, they designed the pilot and the contributed in the construction site design, and then water quality analysis. Then we were sponsored by NGL, PWR, ExxonMobil, and USDA. NGL also provided the water source for the project site, and study design. PWR contributed to treatment technology in field operation and study design, and ExxonMobil also provided analysis support and study design support. I want to thank you, all our collaborators, my advisor, Dr. Thomas Borg, Dr. James Rosenblum at Colorado School of Mines, who took a massive undertaking of designing the treatment plant and running it and doing all the water quality analysis, Mr. Reinhard for his support throughout this project, Dr. Aaron Redman for his support, Dr. Emily and Mike Wilkins, who provided support for microbial analysis, Dr. Calvin Trostel at Texas A&M, who provided support during the planning of agricultural study design, and Mr. Larry Turnbow, who's a farmer and who has been instrumental throughout our time at Orla for growing our sorghum Sudan grass. And lastly, before I close, a quick note, I would like to use this opportunity to let you all know that I'll be defending my doctoral dissertation soon. So my background also includes bachelor's and master's in environmental engineering. I'm exploring roles after graduation. So if you know of any opportunities, I'd love to connect. My contact details are on the slide. And with this, I'm open for questions.
Great. Thank you so much, Huma. You must have stirred up quite a lot here because we have tons of questions here in the chat. I had a question, but I think there's just so many that I'll push mine to the end. And I see Melissa's already got her hands up. So if you're ready, we can kick off the Q&A. And Melissa, you're first in line.
Yeah, both I and Thomas, my advisor, are going to be present to answer the questions.
Awesome. Thank you, Zach. And thank you, Huma, for this great presentation. I just wanted, it flashed so briefly on your screen. Could you repeat the conclusion? I think it said that treated produced water can be used for successful crop irrigation. Is that correct? Is that your conclusion? Okay,
Yeah.
thank you.
Yeah.
Okay, and this conclusion is based on, please remind me, is it just this study alone, or are you referencing other studies in addition to this one as well? Or
So
using other studies?
Thank you. So all the previous studies, the greenhouse study, except for one, we used untreated produced water. That was just to see the risks and to do the risk assessment and to find out the impacts. With only this study, we used adequately treated produced water. So this conclusion is based on this study, provided the fact that this was sort of a pilot. We did it only for one growing season, using only one crop and one soil type. So as I said, that it should be, we were pretty confident about our results, but it also should be taken with a grain of salt that there should be future studies on the large scale, multiple growing season, different crops, different soil types.
Thank you. And just, I had a couple questions in the chat as well. At the beginning of your presentation, you pulled up it was the water treatment train schematic slide. Thank you for whoever's moving that. And it said that, you know, produced water treatment resulted in fresh water. There it is. Thank you, after polishing. And I was wondering how you are defining fresh water here and how that might be different or similar to how the public might define fresh water.
So we use these technical terms in water treatment field that can differentiate it from raw water and brine water. That's a product water that comes out of the polishing step by the end of the treatment. But I can see that public would assume fresh water would not be a treated produced water. And also we have Kaylee from Colorado School of Mines. Their lab oversaw all of this part. So if Kaylee, you want to chime in as well for this question. I think we might not have it.
I can also come in a little bit here. I mean, does the definition of fresh water is in general any water with less than 1,000 milligrams per liter TDS. But I think if you look up freshwater definitions, I think many times you will see naturally occurring water that is not salty is the definition. So it doesn't include brackish water and seawater. But for example, it's common to see that desalinated seawater can be turned into fresh water because you get it to below 1,000 milligrams per liter. USGS defined fresh water as anything below 1,000 milligrams per liter. But I think in general, the general public who say naturally occurring water with a salt concentration less than 100,000 ppm. Some refer to produced water as fossil water. This naturally comes from the underground. I think there's a little bit of debate about whether it's truly fresh water or not, but there's so many definitions. I'm wondering what Pei Su thinks about that. I don't think necessarily we should refer to it as fresh water, just treated produced water that meets the criteria for reuse in agriculture.
Yeah, I agree.
Thank you both. I appreciate that. Thanks for chiming in, Thomas. I had one more question. It was about the bulge effect, but I don't want to just consume all of the time. So, Zach, are there other people? Can I ask about the bulge effect, or should we save that and give other people a chance?
You've got the stage right now, so ask your last question and then we can get other people engaged.
All right, thank you. I appreciate that. Okay, Huma, you mentioned at the beginning the bulge effect of wastewater injection.
Yeah.
Can you describe a little bit more, like exactly what happens with that? And then also the second part is, does fracking, which is also a type of injection, contribute to bulging as well? Thank you.
So, my understanding, Thomas, if I'm wrong, please correct me. Bulge effect happens when we inject at the shallower depths. So deep injection happens at the far deeper depths. And bulge and blowout phenomena, it's the limitation or the function of limitation of the pore space in the soil. So there is so much you can put it in before it runs out of the space to hold that water, and then as a result it causes bulge in the earth and this blowout phenomenon. But it happens at when we inject at shallow depths.
Thank you.
Pei.
Okay, great. Hi, Huma and Thomas. Great work. It's really, very impressive to see such the first field irrigation experiments using the treated produced water, and it's also great to see that the barren soil, were able to grow that the such a green Sudan grass, and also capture birds and also the small animals. I have a question is, I know that James and you have done a lot of work on the produced water characterization, and the treated produced water has a very low TOC, less than 1 milligram per liter. I'm just wondering, did you do any targeted analysis or non-targeted analysis related to the organic compounds in the soil and also in plants? Because Thomas, you have done so much non-targeted analysis, so I'm just curious to see if you will also do the non-targeted analysis and maybe other targeted chemical analysis on the soil and the plant samples you collected.
So yeah, for water there has been in-depth chemical and toxicological characterization, including the non-targeted analysis. But most of the constituents were non-detected under the detection limits, and we did not see. So what our plan was that if we saw something in the water, then we will go in and do the non-targeted for soil and the plants as well. So when we did not see it to begin with, then we plan not to go with non-targeted. In one of the previous greenhouse studies, we did it. But here, because the water quality, like of the treated or the product water, was so good that we did not feel the need to do it.
Yeah, and in addition to that, there was a question in the chat about PFAS. We looked at PFAS. We looked at hundreds of different targeted compounds and quantified them all using a third-party lab, Eurofins, more than hundreds of compounds, and most of them were below detection limit. We also have non-targeted analysis in the works that is led by James Rosenblum.
Right. Yeah, great. Thank you so much, Huma and Thomas. I will let others to ask questions, but I also have more questions, so I can ask maybe later or by email. Thank you.
Thanks, Pei. Tusita, you had a question? Or I can just read it. Let's see. It says, Huma, you may have mentioned in the slides, but what is the statistical test you use to calculate the significant difference?
So we use ANOVA and then post hoc Chucky tests. Yeah, does that answer the question, or?
I believe so. We'll keep moving on for now. Oh, yep. She says yes. Thank you.
Okay.
All right, we had another question. Any comparisons between the freshwater and then parentheses city water and the treated produced water except the TDS?
So they're asking about the comparison between.
Between city water, yeah, potable water.
Yeah, so these are just like, this is just an overview of what we compare, like, but here even there were like pH, alkalinity, TOC, and chloride, and then for these parameters as well. But then due to time constraint, we cannot show the whole data set. It was like hundreds of compounds we looked at, the differences between the city, like fresh water and the treated produced water.
But
And these are just like significant from irrigation standpoint, so I thought to include only these.
But we did the same analysis for the fresh water as we did for the treated produced water.
Yeah.
So we have a comparison, but we did not include it in this talk.
Great, thank you. So I see Amigos Bravos, you have your hand up. Let's do one more that was in the chat, I think before your hand was raised. So we'll just kind of keep the order. When the study comparing the freshwater versus the produced water was run, what was the application rate that was applied on the crops?
So I cannot tell the top of my head. We were doing irrigating on alternate days. But yeah, I need to look at the exact rate at which we irrigated.
But
Thanks, Hum.
I can just add that we had soil moisture sensors in, and so we basically irrigated based on the agricultural needs. So we irrigated just like any other farmer would irrigate based on soil moisture. So we had sensors in 2 different depths, so we could always keep an eye on the water holding capacity and then irrigate it according to the need for the crops.
And because we had access to a lot of water, so we were at the upper limit of we kept the crop happy.
Very nice. All right, Amigos Bravos, you're up.
Thank you. This is Sarah Knope from Amigos Bravos. I couldn't change my name in the chat. Thank you all for this presentation. I have 2 questions. I think they're short. First of all, this is related to the question that Pei was asking, and I just want to make sure that I understand how you were testing the treated produced water. How many constituents were you testing for?
I see more than 300. Thomas, do you know the exact number? Because James Rosenblum at Colorado School of Mines is overseeing that part. But what I see.
Yeah, I just looked at the spreadsheet, and the spreadsheet
Yeah.
indicates that we had more than 310 individual constituents included in our analysis, most of them being organics. But of course, we had the whole metal range, all the metalloids, all the radionuclides, all the nutrients, and then basically all the organic constituents that you can pay for to get analyzed at a third-party lab. So about 310 different constituents. All of them exceeding, I mean, I think any other studies have been done so far. That does not include the non-target analysis. This is just target analysis. Target analysis is more than 310.
Okay, great. Thank you. And are you all, I missed this too, are you all publishing that list of constituents that you tested for?
Yes, I assume that paper from Dr. James Rosenblum is going to be submitted pretty soon. That is one of the reasons we didn't want to show that data as well. So maybe in the next couple of months, and then after that, we will publish our agronomic part.
Okay, thank you. And if I may ask one follow-up question, Zach, would that be okay?
Go for it.
Thank you. Yeah, and as my second question is, as the researchers, did you all ask for and receive any data from the treatment company on a little more detailed on the treatment process? Like, I'm curious how much how much energy it required to treat water to this level, and they're related to that is maybe the cost of treatment. When you all do your projects, do you ask for that data and do you receive it from the treatment companies?
Dr. James Rosenblum also owns that data and company as well. Some of it is proprietary, the pre-treatment part, but the rest, I think they're going to include all the details in the paper they're going to publish very soon.
Okay, thank you very much. That gives me a better picture.
Thank you.
Great. Thanks, Sarah. More questions for you, Huma. Pei asks, will the detailed water quality analysis on city water and treated produced water be reported in future publications?
Yes, very soon.
Awesome. Looking forward to that. And then Dr. Zhang, Dr. Yanyan Zhang has asked, I'm curious what the acceptable boron limit in the soil is for irrigation. What is the concentration in milligrams per kilogram? And does the soil accumulate more boron with time?
Roughly, the ideal is less than 1 milligram per liter, but it also depends on the soil tolerance, crop tolerance as well. So this was higher than most of the crop tolerance, what we saw in the treated produced water. And yes, it does accumulate over time in the soil.
Alrighty. I see you wish you have your hand up. Sorry, one more question in the chat before we get to you. But Subanki, one of the postdocs at NMSU, was saying, hey, great work. Did you measure soil moisture content? And is there any differences among the treatment? And then lastly, did you supply the same amount of water for all treatments?
We did not measure soil moisture content. We did not look at it.
We did measure moisture content. We have soil moisture sensors.
Yeah, through soil moisture sensor, but by the end of the experiment, when we collected the samples and we transferred them all the way to Colorado State, so those samples we did not measure. But yeah, we had a real-time soil moisture data from the moisture sensors, and we are going to include all that data in our publication as well. We already have the graphs, everything ready.
Very good. All right, Wish, you're up.
Hi, Huma. Hey, great presentation. This is really good. My question is actually may or may not relate to what you did. Maybe it's a little bit more on the water treatment side, but you do mention that you want to carefully monitor what the pre-treated water or the treated water needs to be able to assure that, that the soil and the crops are going to be okay. So do you have any inkling on what the particular components that would be problematic, that if the treatment had a breakthrough, what would happen? Or are you just going to go by, hey, everything has to be clean?
Yeah, I think we have, like, the reason we were doing these multiple greenhouse studies and this field study as well, to come up with those problem trouble maker constituents and things that can go wrong and how it can impact soil and crop. So with produced water in our study, as we also noticed that initially we wanted it to be clean, but then we ended up with higher boron and ammonia, not too high, but it did happen. So I think with our studies, we can just guide the regulators and decision makers to make sure that these constituents do not exceed the levels for crop and soil. But I think more trials should happen before it can go out in the field in the real life, just to make sure. And we can also maybe introduce some proxy sensors, which can just can give us real-time values of pH, EC, that if anything is going wrong in the product water, if those proxy indicators exceeds, we know that we should stop and look for what has gone wrong. Yeah.
Yeah, one quick thing, Zach, if that's okay. The reason I ask is because you used membrane desalination to do this. That can only be used cost effectively up to a point, depending on the TDS. What can be used for seawater desalination is not common for what we are trying to do here. So depending on the type of desalination techniques, the constituents that are going to be in the final water could be very different. And so in some ways, it's better to know which ones are more dangerous than others. So yeah, those studies will be important. So you find a cost-effective solution at the end of the day,
Yeah. From irrigation standpoint, I think boron is the trickiest to deal with because of its usually present in boric acid form, and due to neutral charge and then the size, it is even with RO. Sometimes it passes through it. Yeah, but will it be economically viable with RO? This beyond the scope of my pay grade to answer that question. But yeah, it's going to be. But if it comes down to it, and managing that produced water is more expensive than treating it and reusing it
Right.
for that purpose, then we can have a cost benefit analysis. And we in our lab just this year had a paper out which included some of that cost benefit analysis from my colleague, Dr. Marin. I can share it in chat as well.
Thank you. Thank you very much. Great presentation, by the way. Thanks.
Thank you so much.
All right, Huma, we have two more, and then that's probably it, and you're going to be off the hook. Second to last question here is, do you know how much particulate and oil was removed from the produced water prior to receiving the water for use? So I'm assuming, what was the performance of the pretreatment?
I cannot answer that question because, yeah, we did not oversaw that part. But I know that by the end of the treatment, we had drows and grows were non-detected.
Great. And then Pei has one last one to wrap us up. Just for clarification, ammonia of 10 to 11 milligram per liter as N was in the treated produced water?
Yes.
Okay.
We actually have one more question, Zach, in the Q&A. And it says, can you please revisit the different uses for produced water, particularly treatment versus reuse?
Different uses, like they're talking about this.
Daniel Harris, if you would like to unmute yourself and maybe ask that for clarification.
Yeah, I just missed the proportions at the beginning. It was during the introduction where you were talking about how produced water was dealt with right now.
Oh, so 85% is injected back into the ground. 13% is treated, and then less than 2% is reused at the moment in the United States. I can show you the numbers. I'm sorry. So I assume you're talking about these numbers.
All right, thank you. And that reuse is reused in the oil production?
It is also, because in some states in California, it is used for irrigation as well.
Okay.
Yep.
Alrighty. Well, you've done it, Huma. You made it to the end. So thank you very, very much. So very sincere thank you for sharing your time and your expertise, and for everyone joining today. Before we wrap up, we just ask that you please complete a quick survey to share your webinar experience with us. You can either scan a QR code that might be somewhere, or we're going to send it out via email. And so in that email, we're also going to have the presentation and the recording that'll be posted on our website. And so just, we'll be pinging everyone to stay up to date on our next monthly webinar. So with that, we'll conclude, and thanks again for everyone.
Thank you so
Bye.
much.